Hello everyone. Today we are starting with a new topic in paper 3 of T.Y.B.Sc Microbiology, that is Enzymology. The topic which we are starting today is Principles and Methods of Enzyme Purification. Our topic for today is Introduction to Purification of Enzymes. Learning objectives. After seeing this video, the students should gain a concept on the need of purifying proteins and enzymes as well as the approaches taken for any purification procedure. Introduction. Let us see about some general properties of enzyme. So enzymes are biocatalysts. They alter the rate of reaction. Enzymes are very substrate specific in nature. Most enzymes are proteinaceous in nature and are composed of amino acids. Enzymes have a prosthetic group attached to it. Enzymes follow or obey the laws of enzyme kinetics. Enzymes participate in a number of metabolic pathways, and the enzyme action is regulated by presence of specific inhibitors. So where do we get these enzymes from? We can extract these enzymes either from the whole organism or specific tissues or organs where these enzymes are synthesized. A very popular source of enzymes for industrial application is obtaining them from microorganisms. Nowadays, tissue culture is also gaining popularity for production of enzymes in mammalian cell lines. Protein expression systems can also be used. In this, the genes for the specific enzymes are cloned into a specific vector. This vector is then transformed into a living host. The living host now has the property to synthesize the particular enzyme for which is lacking in the host. Thus, can substantially increase the production of enzymes. Industrial enzyme production has evolved very rapidly in terms of both volume and quantity. This is mainly due to the expanding market. There is always an increase in demand for new enzymes which have established their potential application in various industrial structures. Microorganisms constitute the largest and the most dominating source of enzymes. Although many plants and animal sources are also being analyzed for production of enzymes with commercial importance. Production of a microbial enzyme initially involves a screening process in which microorganism with desirable characteristics is screened. The level of natural enzyme production in an organism is often quite low. Hence, it is essential to elevate these levels to meet the industrial demands. This can be achieved by optimizing the media and the culture conditions. This can also be achieved by causing specific mutations in that organisms. Microorganisms used for enzyme production are grown in bioreactors or fermenters. Both solid state and submerged fermentations can be used for industrial production of enzymes. The enzymes are separated from the microbial culture using either centrifugation, filtration, or chromatography techniques. They are then stabilized to maintain a suitable shelf life. Hence, production of enzymes and its purification is of great commercial importance. Enzymes can be classified either as intercellular enzymes or extracellular enzymes. The intercellular enzymes are those which are produced inside the cell and used inside that particular cell itself. They involve many enzymes which are required in metabolic activities. One example is the respiratory enzymes involved in the mitochondria of the cells are produced intercellularly. The other types are extracellular enzymes. As the name suggests, they are produced inside the cell but secreted out externally. Examples of such enzymes are digestive hydrolytic enzymes such as amylases, lipases, which bring about breakdown of large macromolecular structures such as carbohydrates and fatty acids into smaller components or fragments which can be then easily be utilized. Enzyme purification. Enzyme purification is an essential step for characterization of proteins. The purification of protein of interest is an indispensable first step in a series of studies aimed for exploring protein function. Proteins can be separated from one another on the basis of solubility, size, charge, and binding ability. When a protein has been purified, its amino acid sequence can be determined. The strategy is to divide and conquer, that is, to obtain specific fragments which can be readily sequenced. With the advancements of techniques such as X-ray crystallography, we can determine the structure of protein, the amino acid sequence in the active sites, where it will bind to the substrate, what will happen if an inhibitor is added. Purification also helps us to study the kinetic properties of a cell with a better precision. Hence, the study of enzymes and their function is vital from the viewpoint of understanding how the cell's metabolism as well as kinetics work. Enzymes, as we know, play a role of catalyst in many industries. Hence, there is a need to purify isolated enzymes as they have a number of applications which include use in food and dairy industry, use in the paper and pulp industry, use in agricultural industry, use for production of biofuels. Enzymes are also used in textile industry, in pharmaceutical industry, as well as brewing and wine industry. The degree to which an enzyme has to be purified depends on the end use of that particular enzyme. If the enzyme is to be used as a washing powder, then a relatively impure sample can be used. On the contrast, if enzyme is used in food or pharmaceutical industry, high level of purity is very essential. This can be achieved by several purification protocols carried out in a sequential manner. Each of the steps is designed in such a way that it brings about fold purification. But when we are trying to purify proteins or enzymes, it is almost always associated with loss of the product. So an ideal enzyme purification strategy will be the one which has the highest level of purification and the least number of steps. Also, one should be able to minimize the product losses associated with purification. The selection of steps in purification protocol depends on properties of target protein. Here are some essential components which are required for purification of enzymes. Enzymes are mainly used in enzymatic assays. A major essentiality of purification is presence of a source. There must be a judicious source from which the enzyme can be extracted. This source should be cheap and easily available throughout the year. Assay. Assays are quantitative determinants of a particular activity which is present in that particular protein within a particular fraction. One can carry out a diverse range of assays such as colorimetric assay, which is based on changes in absorbance. One can perform assay for determination of molecular weight of protein, like you can use SDS page in this technique. Ability to stimulate cell differentiation, cell proliferation and cell death can also be used as parameters for assay. Since an assay is required to be repeated at least 2 to 3 times, it is essential that the assay procedure is quite simple and not very time consuming. Scheme of purification of proteins or enzymes. In the step 1, we have to prepare a crude extract. This crude, the cells, depending on their nature, enzymes can either be extracellular or intracellular. Extracellular enzymes are secreted out into the fermentation media. This medium is then subjected to centrifugation. After centrifugation, the cellular biomass or pellet is removed, whereas the supernatant is known to possess enzymes and proteins. In case of intercellular enzymes, as the enzyme is generated inside the cell and remains there, cell lysis is an essential step. After bringing about cell lysis, the lysate will contain proteins from the cell cytoplasm, carbohydrates and intercellular components, nucleic acids, as well as cell debris. We need to remove all these components to get a soluble fraction. The second step is the recovery of protein of interest from the supernatants, that is, enriching the protein sample. In step 3, the concentration of protein fraction is concentrated further. This can be done by subjecting the sample to chromatographic purification procedure so as to get even more concentrated factor, one can also use precipitation techniques such as use of precipitants like ammonium sulfate, organic solvents, or polyethylene glycol. The partially pure fraction may then be subjected to one or more purification strategies until purification is achieved. The basic scheme for purification is shown in this slide. We have to grow the cells in a suitable medium. After the suitable growth is obtained, you need to centrifuge this mixture. You will either get a filtrate which is known to contain extracellular enzymes, and you will get a pellet which will contain intercellular enzymes. So, if you want to extract intercellular enzymes, you need to bring about lysis of the cell, again centrifuge it, separate the cell debris and use the supernatant as a source of your intercellular enzyme. After this you get a soluble fraction. The soluble fraction will contain not only proteins but nucleic acids as well. So we need to remove these nucleic acids so that we can get a fraction which is containing the protein of our interest. The extracellular fragment can be used directly as a source of protein fraction which may contain the protein of interest. The next step, as we talked earlier, is concentration, so that you get a enriched protein fraction. Then you try various purification strategies so as to obtain a purified enzyme fraction. Then the enzyme is characterized and can be used in many industrial applications as well. Purification strategy. So when we plan anything or plan a strategy, we are trying to simplify the process. In this case, we are trying to simplify the process of extraction and utilization of resources so that maximum yield of enzymes can be obtained. We mainly use a 3-step protein purification strategy. It is known as the CIPP, or the capture, intermediate purification and polishing strategy. So this CIPP strategy can be used on laboratory scale as well as on industrial scale. It mainly ensures a very fast process as it requires short time, and also it is economically favorable. So in the capture phase, we have to isolate, concentrate and stabilize the target enzyme. The next phase is the intermediate purification phase. In this phase, we have to remove the bulk impurities from many other unwanted impurities. These unwanted impurities can be other proteins, nucleic acids, endotoxins and very closely related structures which are not removed. Polishing phase. In this phase, the most major impurities have been already removed. So this phase mainly focuses on removal of any trace or specified impurities or unwanted proteins which are formed during the reaction. So as you can see in this graph, the purity increases as step by step you are performing extraction, capture, purification and polishing. Although this strategy consists of a broad phase, it does not imply that every purification process should contain only 3 phases or 3 steps. Each of the phase, for example, capture phase may contain 5 steps. In some other capture phase, it may contain 2 steps. In this intermediate phase will contain 2 to 3 steps depending on what enzyme you are extracting. So depending on the specific purpose or a specific problem, a specific purification step may be addressed. So number of purification steps primarily depend on the intended use of the protein. However, it should be kept in mind that as the number of purification steps are increased, the overall protein recovery and activity will decrease. At the same time, purification time and cost involved will go up. The solution to this is optimizing the purification strategy so as you get the maximum yield of purified protein and fold purification. So as I stated earlier, enzyme purification is a multi-step procedure. We'll talk about it in a step-by-step manner again. So first you have to collect the sample, then apply any purification technique, fractionate this sample, then we have to assay for total number of proteins present in that sample. After that we have to assay for enzyme activity of that sample. If the enzyme activity is not up to the mark, then we'll set it aside, and if the enzyme activity is up to the mark, then we need to monitor its purity by performing SDS page. If we obtain a pure sample, then we need to proceed for characterization, whereas if the sample is still impure, then combine all the impure fractions to concentrate them and repeat the purification procedure until you get a pure protein or enzyme. So when we talk about steps of enzyme purification, the first step is solubilization of the enzyme. One can either use a homogenizer, a centrifuge, or filtration technique to obtain a filtrate. After obtaining the crude enzyme, the enzymes in the mixture need to be stabilized. After stabilization, we need to isolate these enzymes from the mixture and store them under appropriate conditions. Some biological materials themselves constitute a clear or nearly clear protein solution suitable for direct application to chromatography column after centrifugation or filtration itself. Step when we consider purification is homogenization for solubilization of proteins. It can be carried out on both laboratory scale as well as industrial scale. For homogenization, one can either use sonication, French pressing, enzymatic or chemical cleavage of the cells, freeze thawing, and for industrial scale, one can use either high pressure homogenizer or bread milling. A substitute to homogenization is centrifugation. In centrifugation, one can isolate the enzyme components and separate them from the membrane's cellular and subcellular assembly by subjecting it to rotation under high speed. The cell lysate is centrifuged at a high speed to remove the denser components of the cells, which settle as a pellet at the bottom of the centrifuge tube, whereas if we are performing it for extracellular enzymes, the lighter components will be in the supernatant. The second step is stabilization of the enzymes. There are many factors which affect the stability of the enzymes. The first one being the pH. So to prevent denaturation or loss of function, the enzymes are mainly placed in buffered solution. The pH is near native pH of the enzyme. Temperature also plays an important role in stability. Purification of enzymes is mainly carried out at a low temperature ranging from of approximately 0 degrees Celsius. Some proteins are thermally stable, so purification can be carried out at slightly higher temperatures. We also need to take care that no proteases are present in the medium which will bring about destruction or cleavage of the desired enzyme. So the third step is isolation of the final product. You can use a number of methods and utilize many properties of the enzyme for its isolation. The first characteristic being the solubility of the enzyme. One can use either ammonium precipitation or salting out methods. One can also make the use of difference in molecular sizes, and we can use techniques or procedures like gel filtration chromatography, dialysis. Difference in ionic charge can also be used for separation. For example, use of ion exchange chromatography. Separation based on binding affinity to the column, that is affinity chromatography. Separation based on hydrophobic interactions of protein with column matrix, that is hydrophobic interaction chromatography. Fractionation by isoelectric focusing or any other method available. So fractionation or isolation will mainly serve two purposes, that is removal of added contaminating material as well as enriching the protein fragment of interest. As soon as the fractionation is accomplished, the fractions are assayed for their activity of interest. This activity will only be present in some fraction, and this fraction will be containing the target protein. These fractions are then quantitatively analyzed using tools like sodium dodecyl sulfate page. If it is not completely pure, the enriched fraction containing protein of interest but not completely pure is further put through another round of purification, and the process is repeated till a pure protein is achieved. So thank you so much.